I'm going to use these minutes that I have to tell you about how to surpass the sub 10 nanometer resolution limit in fluorescence nanoscopy. And so let's get started. So before going into the science, just a few words about where I come from. So we work at the Center for Bio-Nanoscience Research in Buenos Aires, Argentina. And we are a small group of researchers. Here you see a few snapshots from a few years back. Here more recent pictures. And so we work on different subjects, but what I will be telling you about today was mainly the work done by Luciano Masullo, Lucia Lopez, and Alan Salai. And well, Luciano is and Alan are already in postdoc positions, both in Munich actually. And now the group has this configuration here with Santiago Sosa, Luciana Martinez, Lucia Lopez, Yanina Violi, Gonzalo Escalante, Florencia Choque, Cecilia Sasa, and Florencia Dorma. Okay, I'm going to skip this. These are other experiments that we do. Just a few words about DNA origami because we are going to use it during this talk. So for those of you who don't know what it is, DNA origami is a technology based on DNA. I'm sure you all know DNA. You know, it's a biomolecule, it's a biopolymer that encodes the genetic information of all living organisms. But here we are not going to be dealing with the biochemistry or biology. We will use DNA just as a special molecule to fabricate nanostructures. So DNA has a very particular property that it can form a double helix, a double strand. And it does so only if the right sequence, the complementary sequence is present in the two chains. So imagine that you have a single-stranded DNA, like this one here, very long, but you know the sequence in all this chain. And now you incubate this single-stranded DNA with a small chain of DNA, an oligonucleotide, that is complementary. Half of it is complementary here, like green with green, and the other half is complementary here, like red with red. If we let this hybridize, and we let DNA make this double helix, this polymer chain will fold in this way, in this region. So to do that, you need to know the sequence of both chains, of course. Now, if you design many small chains with the right sequence, then you can fold this polymer into a precise geometry, like you see here. This is what is about DNA origami. And it's called origami because you're basically folding a DNA chain into a predefined shape. So this can be done. What you see here is an AFM image of zillions of those rectangles self-assembled by DNA hybridization. So this is interesting for us because you can use these structures as like nano breadboards, like nano structures where you can place other things on them with a defined geometry and stoichiometry, and recently actually also with defined orientation. So what you see here is an example of how we use DNA origami, in this case a 3D structure, to hold two nanoparticles and one fluorophore, that's the red dot here, to generate a unidirectional optical antenna. So we are using this origami for nanophotonics, but also, and this is what we are going to be seeing today, is also for calibration samples for super resolution microscopy. Okay, so let's get to the topic of today. So we are a small group, but we are lucky to have a strong network of cooperators all around the world. And the work I will show today was done in collaboration with the MPI in Göttingen, Stefan Hell and Tom Jovin, the INIMEC in Córdoba, Alfredo Cáceres, Nikon Sain, Mariano Bisbal, in Buenos Aires, Ibiova, with the group of Damián Refojo, in Munich, with the group of Philip Tinefeld, and Fribourg with Guillermo Cunha, and in London with Sabrina Simoncelli. All right, so fluorescence microscopy. I don't need to explain this. You already know how great it is to visualize biological structures and other structures. But its resolution limit is restricted by diffraction of light, by the wavelength of light. And that is basically because when you focus light, what happens is that you have an interference phenomenon. So when you build an image from each point, what you get is basically an image where all the waves converge in phase in one point, and then you have a maximum of intensity. But that maximum of intensity has a finite size determined by the wavelength of light. And for that reason, even if you start with a point-like light source, you end up with a signal that is more or less half the wavelength in this dimension and one wavelength in the axial dimension. That's the resolution limit. I show this example because we don't have point light sources, like mathematical points emitting light do not exist, but molecules, single molecules are very small, could be 1 nanometer in size, and they emit light, fluorescence light. And if you make an image of them, you will register a signal with a size of about 200 nanometers. So you see, you start with something of 1 nanometer in size, and you end up with a signal 200 times larger. This 1 to 200 mismatch in size is basically telling you how much information you're losing when you do diffraction limited microscopy. And this is another example I usually give. 1 in 200, if you look at a football stadium, a human being has a size of about 1 meter. The football field has a size of order of magnitude of 100 meters. So this is 1 in 100. If you would look at a football match with 100-meter resolution, there's no way to tell where the players are, where the ball is. There's no way to tell even there is a football match going on here and people watching it around it. So that's how much information you lose when you do diffraction limited microscopy. So people have tried to tackle this, for example, by colocalization measurements, but it's the same problem because if you would do this kind of measurement here, in this case, the Netherlands and Argentina are colocalizing in this 100-meter field, but there's no way to tell what's going on inside there. So luckily, this was solved already a couple of decades ago by fluorescence nanoscopy or super resolution microscopy. You probably all know about it. There are different families of techniques like coordinate targeted methods like STED. These are applied in scanning microscopes, or coordinate stochastic methods that are applied in wide field microscopes like STORM, PALM, DNA paint. Any of these methods provide a much higher resolution, as you can see here in these examples of microtubules in cells. Here you see when you apply STED, the resolution improves dramatically. The same here on the right. So these methods really allow you to see more. And actually, they enable the discovery of structures that were invisible before. This is a great example from the lab of Xiaowei Chuan, and they discovered this regular structure in neurons called the membrane-associated periodic skeleton that is made of different proteins. And you see this very regular structure. This is on the right, an image from our lab using a different technique, STED in this case. And you see, if you would make like here on the upper right panel, if you would make a diffraction limited image, you would think that these proteins are uniformly distributed in the neuron. But in fact, when you apply super resolution, you find out that these proteins are not uniformly distributed, but rather organized in a very regular pattern, like you see here. So super resolution microscopy is really a breakthrough, and this motivated the Nobel Committee to give the Nobel Prize in Chemistry to these 3 gentlemen here. But it has so how does it work? Let's see very briefly the concept of how does it work. There are two family of methods, we say. The first one is coordinate stochastic, and it works in this way. Imagine you have, like you see here, molecules, fluorescent molecules, organized in some pattern. Let's say like this smiley face here. But the distance between the molecules is smaller than the diffraction limit. Then, if all these molecules emit at the same time, you will register a signal like this one here. Just a blob of bright signal, and you cannot tell where each molecule is. But if you have some way to switch these molecules off, and then switch on just a few of them, separated enough, so that you can, then they are, you can register signals of individual molecules, not overlapping. Then these signals, of course, will be diffraction limited, but if they can be assigned to a single molecule, then you can determine the center of that signal and tell, okay, that's the position of one molecule. And here I have another molecule in this position. And now I switch them off and I switch on other molecules. And I repeat the process and determine their position. And this can be repeated over and over again. And at the end of the day, you end up with a list of positions of molecules. And with these positions, you can reconstruct an image, but it's no longer limited by diffraction, but it's limited just by the error with which you determine each one of these positions. Okay? So based on this concept, there are many methods depending on how you make these molecules switch on and off. And the most known are storm, palm, or paint. Okay, and here you see an example. So in practice, you do not register an image, but a video. And then you see blinking molecules switching on and off. And then in this case, the blinking is due to DNA hybridization. Basically, this example is here, a DNA origami with 3 docking sites where fluorophores that are swimming, fluorophores linked to DNA that are swimming in solution can bind transiently to these docking strands. So they remain here for 1 second or half a second, and then they go away, and then comes another one, and then comes another one to a different position, and so on. So what you have to do is then analyze the video, each frame of the video, detect those signals, determine the center, and then reconstruct an image. The error with which you determine the center is actually inversely proportional to the square root of n, and s here in the numerator is actually the size of the signal, the diffraction limit. And if you do that, you see that that video actually corresponds to an image like this one, where each bright dot is in fact actually coming from 3 dots in line, just as the DNA origami design was. Okay, so you can super resolve by doing this. And of course, it can be applied in biological samples, like I showed you before. And the resolution here is limited by the error in the position determination, which follows this law, which is basically the inversely proportional to the square root of the number of photons. And so, to summarize this method, what you need is homogeneous illumination. So your sample is illuminated all with the same intensity. And then you localize molecules by using their emission light. So all the information is extracted from the emission of the molecule. That's very important. Let's switch now to the other family of methods, that is coordinate targeted. In this case, we are going to measure one position of the sample at a time in a scanning microscope. So now again, let's imagine we have that sequence of molecules, but the distance between them is smaller than the diffraction limit. Then, if I record a fluorescent signal with the scanning microscope, it would look something like that, because the focus of light, the focus of the excitation light is larger than the distance between the molecules. We will register a quasi-homogeneous signal. Now let's imagine that we have a mechanism to switch off molecules, again on and off switching. But in this case, the off switching has to be induced by light. So imagine we have some mechanism to suppress fluorescence or to switch off fluorescence using light. If that mechanism exists, then we can superimpose to the excitation focus another focus of light, but in this case, a special focus that instead of having a maximum of intensity in the center, it has a minimum of intensity in the center, ideally a zero of intensity. This can be done in different ways. We can talk about that later. But basically, you need to make the waves reaching the center to arrive in antiphase. So they, instead of making constructive interference, they make destructive interference. And then you get a minimum at the center. Okay. So if we have these elements, this donut-shaped focus and a mechanism to suppress fluorescence, now we scan our sample. Now the region of the sample from which we record fluorescence is smaller. And if we can do that more dramatically, eventually, we will start discerning where each of these molecules is located, okay? So this is another way of breaking the diffraction limit. And again, there are many methods based on this concept. The difference between them is the mechanism, the light-driven mechanism used to suppress fluorescence. The most common of them is STED, which uses stimulated emission to suppress fluorescence emission in a determined spectral range. Okay. So this is a conceptually very different method, because now all the information about the molecular position is injected with the illumination. Remember, in the other method, it was all in the detection. The illumination was homogeneous. Here, it's all in the illumination. So if actually in an ideal situation, I would only need one photon to know if a molecule is here at the center, at this target position, because I could switch on these two beams, wait some time, and if I detected one photon, I could tell with some statistical confidence that underneath that focus there is a molecule, or not, depending on some threshold. And then I can move this to a different position of the sample and ask the same question: is there a molecule or not? In practice, because there is always background and the switching off is never perfect, you will need also a large number of photons to have a good resolution. So both methods, they work in different ways, but they both end up needing a lot of fluorescence photons to achieve a very high resolution. That's very important. So here you see an example of STED, or coordinate targeted super resolution on the trypanosoma cruzi. Okay, and diffraction limits imaging doesn't provide information about the nanoscale organization of these two proteins. When you do super resolution, you detect these clusters very nicely. So these two families of methods actually do not have any fundamental limits. In principle, they could achieve any resolution, but they do not. They do not due to a practical limit. So if we zoom in here, in these microtubules, we will see that everything starts to be blurred again. And the reason for that is that all fluorophores, at one point, they photodegrade, they photobleach. So we have to work with a limited number of photons. And that makes, that imposes a resolution limit that is around 15 to 60 nanometers in the lateral dimension and 30 to 120 nanometers in the axial direction. So this is a practical limit for, to work under ambient conditions in liquid environments, but it's very important if you want to image biological specimens. So we need another push into the resolution. We need to get below the 10 nanometer regime. And why below the 10 nanometer? Because that's more or less the size of structural proteins. So if we could surpass the 10 nanometer resolution regime, then we will be able to image single proteins, the position of single proteins, and in that way start seeing how proteins self-assemble to form higher order structures, like these microtubules, for example, or other organelles and tissue eventually. So move that resolution limit, there was a development of a second generation of fluorescence nanoscopy. That is basically, it works in two ways again. So the problem is the limited number of photons. Fluorophores photo bleach, so that's the problem. If you want to surpass this obstacle, there are basically two ways. You either get more photons somehow, or you get more information from the few photons that you have. These are the only two solutions. So, and both of them have been investigated recently. So to get more photons, DNA paint was developed, for example, but not only that, stabilizing buffers, self-healing dyes, and other approaches. Here you see some reference names on this approach. And to get more information from the few photons that we have, also there were several approaches developed, and the first one was MintFlux, but now I will show you other ways too. That has been also a focus of work in our research group. So we have worked on different ways of surpassing this limit. And so one was step-FRET, the other one was simpler, which is a method that works under total internal reflection illumination. And then this family of methods that we call single molecule localization with sequential structure illumination. I will focus more on this. So I have just now 20 minutes left, so I will go very quickly over these approaches, and probably I won't be able to go into technical details, but we can discuss them further later. So STED-FRET, you all know FRET, I'm sure. STED, we just saw what it is. So this is basically a combination of them. And so I'm not going to explain FRET because I assume you all know about it. And FRET imaging has been very useful now to visualize the interactions and functional states of biomolecules inside cells. But when you do FRET imaging with diffraction limited methods, what you obtain, of course, is the average information over hundreds or thousands of molecules interacting in a diffraction limited volume. And also, it is impossible to tell where exactly those interactions are taking place. So that's the motivation to develop super resolved FRET methods. And in that way, for example, here, this is again this periodic membrane skeleton of neurons. And this is supposed to be a structure like this, where actin filaments in green form rings linked by adenosine in blue, and these rings are separated by spectrum tetramers. And that is what makes this very regular spacing between the rings. So the size of this structure is about, the spacing between rings is about 190 nanometers below the diffraction limit. So here you see an example of this structure here below. So if you do confocal imaging, you cannot even resolve that structure. You cannot tell that structure is even there. Now, if you do STED, like I show you here, then with a 40 nanometer resolution, you can clearly resolve the presence of this structure. But now, if you want to see molecular interactions inside this, you need something more, something like FRET, which gives you information about 5 nanometer distances between molecules. So we want to combine STED to super resolve protein structures with FRET to give us information about biomolecular interactions inside those subdiffraction structures, okay? And this is what we did basically with an intensity-based FRET measurement using STED. So this is a two-color STED microscope with 2 excitation beams and 1 STED beam and 2 detection windows. That's the configuration here. We used the ATO 594 as donor and ATO 647N as acceptor. And you can find further details in this paper. But basically, we again used DNA origami to test that this was working. This was a DNA origami with 2 FRET spots separated by 180 nanometers, and we could clearly resolve it. The state resolution was about 40 nanometers. And now we can do FRET on each super resolved spot. As you can see here, we can see single molecule FRET by these anti-correlated signals. And then, okay, based on a intensity determination of FRET based on a formalism developed by Tom Jobin and Ellie Harris a few years back, we adapted this to state measurements, and there we could really resolve, for example, in here, you see these are two neurons in diffraction limited imaging. You cannot tell. Here we are imaging adenosine and actin, the two proteins involved in this periodic skeleton. In diffraction limited images, you cannot tell where those proteins are, and even less if they are interacting. You detect some interaction, but it's very hard to locate them. When you do state FRET, you can visualize the regular structure, but also you can detect that those proteins are actually interacting inside those structures. This is something that is impossible to do by single molecule FRET or by diffraction limited FRET alone. Okay, the other approach is called simpler, and you find details in this paper. It's a TIRF method. In this case, to obtain direct super resolution below 10 nanometers in 3 dimensions. So it's limited to TIRF, but it gives you below 10 nanometer resolution in all 3 dimensions if you combine it, for example, with DNA paint. This is a method that is to be combined with coordinate stochastic super resolutions, like Stone, DNA paint, or Far. And the idea is basically to calibrate the signal of single molecules. You know that when you do TIRF illumination, the excitation field depends on the axial position, but that's not, so this will make molecules brighter when they are closer to the interface, but that is not all. Also, the emission of molecules changes as they approach the interface. And you see here, Z is the distance to the interface in nanometers. You see that as they get farther away, they emit less light into the glass. That means less light into your microscope objective. And therefore, you have another effect that makes molecules brighter when they are closer to the interface. So if you combine these two effects, you get a good calibration of your microscope. You see here in red is the exact solution, and in blue is an exponential fit. So its calibration is almost exponential. For practical purposes, you can use an exponential approximation. That means you can calibrate your microscope with just 3 parameters. This alpha F and DF can be calculated for an optical system. It just depends on the geometry and refractive indices of your experiment. And N0 is actually the number of photons that your fluorophore gives when it is sitting exactly at the interface. And this can be measured because you just need to throw some fluorophores on your glass and measure it like we did here. With DNA paint, we were getting about 50,000 photons from each molecule. And with this number of photons, your lateral resolution becomes below 10 nanometers, and your axial position is basically determined from the intensity. So what we do is, just as I showed you, we register a video to localize single molecules, and then molecules switch on and off, like you see in this sequence. We discard the first and the last frames because we don't know if the molecule was emitting during the complete time. So we only take the inner frames, and from them we determine an average intensity, and with that intensity we obtain a Z position using our calibration. That's very simple. So with this, calibrating a TIRF microscope, you can transform any 2D super resolution method into a 3D super resolution method. And now you can see, for example, here, these microtubules, we can not only see them super resolved in 2D, but also obtain their cross-section. You see, these are wide set projections, and you can really see the cross-section of microtubules, which is a very small structure. Actually, the resolution here was about 8 nanometers in this experiment. So this is basically a calibration software problem. So we also provide an open source software for anyone who wants to apply this in their microscopes. Okay. Just a break. Are you still there? Yes? I feel I'm going a bit fast. So now we have 15 minutes left. Yes. And I want to show you the last approach, which again involves a family of methods. And we call it single molecule localization with sequential structured illumination. So this, at least for us in our group, this all started with the MinFlux. This is a method that determines the position of a single molecule using a donut-shaped focus, a focus with a minimum of intensity in the center. And how does it do it? Well, it was basically a triangulation problem. So as you can see here, we were exposing the molecule to 4 positions of this beam, not 3, like you would do in a normal triangulation problem. We needed 4 due to the symmetry of our beam. But nevertheless, so we exposed the molecule to 4 positions of this beam and registered the fluorescence intensity at each position of the beam. And from that, we could determine the position of the molecule very precisely, and more importantly, using very few photons. And also, L is like the dimension of these arrangements. We could make it as small as we wanted, because the zeros, you can bring them as close as you want. And therefore, we had a localization error that, of course, it depended again on the number of photons, like it's, of course, done by implied by statistics. But L, now in the numerator, we have L, which we can make as small as we want. So this method allows you to zoom in as much as you want into sub-diffraction areas. And here you see how this zooming in works. This is the localization error as a function of the total number of photons. These black lines correspond to camera-based localizations, and these colored lines correspond to MinFlux using different L. So you see, for example, if we have 500 photons, with an ideal camera, we would get a precision of about 7 nanometers. With a real camera, we would be more in 20 nanometers. But with MinFlux, real experiments, we can get 5 nanometers using L of 150 nanometers. But if that's not enough, we can zoom in, reduce L, and now our precision becomes 2 nanometers. And if that's not enough, we can reduce it further, and then the precision becomes 1 nanometer, and so on. Here you see some localization histograms. You see, using just 50 photons, the localization error is about 10 nanometers. And if you use a few thousand photons, then you already are in the 1 nanometer regime. So you can localize molecules very precisely in this way. So you can do nano imaging, like you see here, or you can do super fast tracking. And this is also compatible with film measurements, because it's easily combinable with lifetime measurements. So experimentally, this was first implemented in this way. It looks very simple. It's like a confocal microscope with just these modifications. Like we needed a drift correction because the measurements take time. So you need to stabilize your sample to 1 nanometer or less if you want to achieve 1 nanometer localization precision. And then you need, of course, a vortex faceplate or some other phase modulation device to obtain your focus, your donut-shaped focus. And then you needed to move your pattern, shift it between these 4 positions as fast as possible. So for that, we used in our first implementation an electro-optic modulator and an electro-optic deflector, like an electro-optic scanner. And this allowed us to do this rather fast at 8 kilohertz. But these electro-optic devices are expensive and they need fast electronics to use them properly. So this setup was really complicated and not many groups were using it around the world. And actually it's commercialized in a few million euros. So a few years later, we came up with this other approach, which looks more complicated, but it's actually simpler. What we did was to take a pulsed laser. We split the pulse train in 4 and then put some delay on each of the beams, like around a quarter of the period, and then recombine them. And recombine them in a shifted position to form this mean flux scanning pattern. So now this pattern is generated at the sample plane at the repetition rate of the laser, much faster, at 20 to 40 megahertz, depending on the repetition rate of the laser. So now you can do these measurements much faster, and all you need is just some mirrors, beam splitters, and optical fibers, which is much easier than the electro-optic devices. So, but now, okay, so mean flux is very efficient in terms of photons and localizing single molecules. And this, at that point, was really a breakthrough. And after that, many other methods started to develop, like Winstead and others. But if you look back in time, other similar methods were used to localize single molecules. For example, one of the earliest, or the earliest I know, was orbital tracking. That was first proposed by the group of Jörg Enderlein, theoretically, and was first done experimentally by the group of Enrico Graton in the early 2000s. Here, you would shift a Gaussian beam, so a beam having a maximum, around a circular orbit to determine the molecular position. Or this other method, called the four-focus particle tracking, is very similar to beam flux, but in 3D, but again, using Gaussian foci, and not zeros of light, but maximums of light. But it's very similar. Or this other called single molecule confocal tracking, which is remarkably was published in Nature, and it's just a reinvention of orbital tracking. It's nothing else. So what I want to show you here is that over the years, several methods were developed to try to localize single molecules very precisely. And these methods were developed independently with different ideas, with different setups, with different data analysis routines. And therefore, they looked very different. But in fact, they can all be understood within the same framework that puts all this together, and that allows us to compare them in a fair way. So all these methods basically require three elements. And basically, what you need is an excitation field that depends on position. So you cannot use homogeneous illumination. You need a field that depends on position. Then you need to shift that excitation field over a sequence of positions that we call Ri. And you have to register, of course, the fluorescence signal at each one of those positions that we call Ni, because we detected the signal in photon counts. Okay, I have my clock telling me I have to hurry up very much. All right, so with these three elements, you can infer the position of a molecule. And you can do this in many different ways. For example, you can do an orbital scanning, or you can do a regular scanning, or you can do this weird mean flux scanning. It doesn't matter. It can all be described in the same way. And if you use focused light, there are basically two options: intensity maximums or intensity minimums, or zeros. That's all. So now you can think of the different possibilities, like orbital tracking, I told you, is using a Gaussian beam and a circular orbit. You can do confocal single molecule localization using a Gaussian beam and a raster scanning, or you can do mean flux using a donut beam and this weird scanning. But also you can do other things. For example, you could think of orbital tracking with a minimum. Why not? Or raster scanning with a minimum. Why not? So from all these, we tested them, and here you can see. And you can find out that all methods using minima of light are very efficient in terms of photons. Well, I don't have time to explain this. So we decided to test experimentally this one, Rasmin. Why? Because it uses raster scanning, like any scanning microscope. And all you need to do is just to scan with a minimum instead of with a maximum. And then you can get to localization precisions as good as mean flux in a much simpler way. So you need basically a scanning microscope, a confocal or two-photon microscope. You need to add a phase modulation to have your donut-shaped focus, and you need to stabilize your microscope. This can all be done, and you find details in this paper. So if you do that, basically, here you see examples of single molecules localized. You can really localize them with 1 to 3 nanometer precision using a few hundred photons. And now you can do imaging, for example. Again, we use DNA origami with 6 fluorophores in this case. They look like this in a diffraction limited image, but what we need to do is just to pick one and scan a small area in the center and let the molecules blink. Here you see the molecules blinking. And as they blink, they looks like that. Here we are scanning repeatedly 100 nanometer area with 6 by 6 pixels. And when the molecules switch on, you see a minimum somewhere. You detect where that minimum is, and then you collect positions, and you end up with an image that looks like this. And you see the scale bar is 10 nanometers, and you can clearly resolve molecules that are those distances apart. Okay. With that, I finish. And here you see an example of how much better you can localize molecules. And I hope you have a nice idea about how this works. And just to end up, a few thoughts about the future of these techniques. I think that there is good potential for fast and accurate tracking using pulsed interleaved mean flux, this very fast mean flux method I showed you. And for nanoimaging, I think these methods are losing relevance when to localize really with very high precision. Why? Because remember, the limitation was due to the limited number of photons. But there were many advances now and tricks to get more photons from fluorophores, like DNA paint, stabilization of dyes, etc. And there's also limitation in the labeling size. So if you really want to exploit a 1 nanometer resolution, you need to label your sample so precisely. So you cannot use nanobodies or, of course, not antibodies. So there's a limit there, a practical limit on how useful these methods will be, at least for the moment. But there is another edge, that is to use bad fluorophores, like fluorescent proteins, that were discarded for super resolution microscopy in the past. We can use them with these methods to get to 10 to 20 nanometer resolution, which is quite decent. And now many more fluorophores can be used. Okay. And then there are other approaches for physics measurements that I can explain later if you're interested. And here you will find a review about these methods that this was recently published in ACS Photonics. Okay, thank you. And I'm